Dr. Madangi Ganapati has obtained her MSc in Genetics and Plant Breeding from Kerala University and PhD from School of Biotechnology, Madurai Kamaraj University. Having a passion for gaining knowledge and for instilling curiosity in fertile minds, she has been active in the field of teaching and research for more than 15 years. Her current research interest includes recombinant protein expression in plants, transgenic plant development and manipulation using molecular techniques, and analysis of changes in basic gene regulatory pathways during transgene expressions, nutraceuticals, and the role in alleviating certain chronic human diseases. She has a number of publications in peer-reviewed international and national journals.
Welcome to the lecture series on food microbiology. This is the second part of the lecture where we are going to deal with natural antimicrobials. In the earlier lecture, we dealt with three bacteriocins, nisin, subtilin, and natamycin. We discussed about their mode of action, how they preserve the food, what is their antimicrobial effect, whether it works against bacteria or whether it works against fungi, and what are the conditions which need to be maintained in the food product if these are going to be used as a preservation. Today we are going to talk about two other antimicrobials which also play a huge role in preservation of food products. The first among them is tylosin. This tylosin is a non-polyene macrolide antibiotic like your erythromycin, olindamycin, etc. As you all know, erythromycin, olindamycin, almost everybody would have been prescribed and would have taken for any sort of antibiotic infection, especially erythromycin. Similar in structure to this erythromycin, or in other words, non-polyene. Your tylosin is also an antibiotic like that. We call it, that class is a macrolide antibiotic. This is more inhibitory than nisin or subtilin, meaning the range of microbes which it can inhibit is potentially more, which makes it more effective. And unlike nisin, subtilin and natamycin, tylosin is used in animal feeds and also to treat some diseases of poultry, meaning because it is potentially more inhibitory and very effective, this is normally used as a part in veterinary medicine, used in animal feeds. So normally when you want to treat animals, you mixed the antibiotic with the feeds and you supply them. Else along with poultry also, this is how we supply. So this tylosin is normally added in animal feeds for treating the animals and also for some diseases of poultry, which means that this is a part of the treating, it's a therapeutic antibiotic. So you have some limitations for using it for food processing and preservation. And the effect of this is more against gram-positive bacterium. Most of the antibiotics actually, because of the peculiarity of the structure of the gram-positive cell wall, many antibiotics work against them, and you have also some limitations there also. And tylosin is something which normally is utilized against gram-positive bacteria. This inhibits protein synthesis by associating with the 50S ribosomal subunit and shows at least partial cross-resistance with erythromycin. So normally, which means that this antibiotic is a potential protein synthesis inhibitor. For protein synthesis to happen, if you remember your basic molecular biology, the ribosome has two units, the small subunit and the large subunit. In the case of bacterium, it is 50S subunit and 30S subunit. Combining together during protein synthesis, this forms a 70S ribosome. The messenger RNA, or in this case, because in a bacterium it is coupled transcription translation, the transcribed mRNA is immediately goes into the translation mode. Okay, so for this translation to happen properly, as you know, the subunits have to unite together, and then you have a space for the messenger RNA to go inside and then undergo the translation methodology. So here, when the 50S ribosome, when your antibiotic goes and binds to the 50S subunit, remember what we are talking about of a 50S subunit, it is a protein, and each protein has a specific binding site which is specific in structure, and this is like any receptor ligand union. So what will happen is that when a receptor and the ligand form a complex, each ligand is specific based on the structure of the receptor. You have the conformational similarity between them. So then when this happens, when two proteins like that unite together, we have what you call as a conformational change. This is a basic biochemistry of any receptor ligand interaction. So when your antibiotic binds against the 50S ribosomal subunit, you have a conformational change in the structure. This change in the conformational structure in turn can prevent it from binding to the 30S subunit and form the full structured ribosome. So non-formation of the full structured ribosome will inhibit protein synthesis, as a result of which the organism will die. So most of the protein synthesis inhibiting antibiotics either go bind to the 50S subunit or to the 30S subunit, or they will prevent the mRNA from binding. So these are the normal methodologies for the effectivity of the antibiotic. So like any protein synthetic inhibitor, this tylosin binds to the 50S ribosomal unit. It associates with the 50S ribosomal subunit, and it actually shows a partial cross resistance with erythromycin. So this, because of these peculiarities, because it is used in the treatment, the potentiality of usage as a food preservator is comparatively less when compared to nisin and saptolin, even though it is more inhibitory than nisin and saptolin. Now let us see some antifungal agents, especially for the fruits. Normally when we talk about microbial spoilage of the fruits, as because of the nature of the fruit concerned, many fungal agents, it's the fungi who destroy the fruits. So we normally use antifungal chemical agents for the preventing the spoilage. You have the thiabendazole, which is usually sprayed against apples, pears, citrus fruits and pineapples. You have the binomyl, which are again used against a series of fruits including citrus fruits. You have the biphenyl, which is used against citrus fruits specifically. Then we use sulfur dioxide fumigation for grapes. Then sodium alpha phenylphenate for apples, pears, citrus fruits and pineapples, etc. So these are just some of the chemicals which are normally used. Like that, you have specific antifungal agents. The other major antimicrobial, natural antimicrobial which is used is lysozyme. This lysozyme is an enzyme which was discovered by Alexander Fleming in 1921. Fleming described this as a remarkable bacteriolytic principle because this was extremely capable of breaking down the bacteria, lysing the bacterial cell. Okay, bacteriolytic. He discovered penicillin only after the discovery of lysozyme. And as you all know, Alexander Fleming was knighted in 1944, and he also received the Nobel Prize in Physiology and Medicine in 1945 for his discovery of the antimicrobials and also for the treatment of infectious diseases. Fleming, as you all know, penicillin discovery was actually accidental, right? He actually saw, he was culturing a series of microorganisms in a petri plate, and one day he observed that there were some fungi which were growing on in the plate, and wherever the fungal germination and hyphae was seen, he saw the zone of inhibition of the showing that the bacteria lysed. Then he said that this was because of some product of the fungi, and this was actually paved the way for the discovery of penicillin. And like that, the discovery of lysozyme and lysozyme research, he emerged from his demonstration that chemical antiseptics were ineffective in treating infections. See, antiseptic is something which you actually use to disinfect outside, you don't take it in. And there were a lot of antiseptics which were used in hospitals, in labs, etc. He showed that these antiseptics could momentarily kill as long as the antiseptic was used. For the time being, you could control the microbial inhibition. But when you have an infection, these cannot be used to cure. So then he was trying to find out what can be used to treat the infection. Then that mode of research actually led to the discovery of lysozyme. He showed that his own nasal mucus inhibited the growth of micrococcus species. Now the question is, if nasal mucus can inhibit micrococcus, then what is the composition of the natural mucus? Is there anything specific in it which can destroy the species? He first thought it might be because the inhibitory factor is a bacteriophage, because as you know, bacteriophages are viruses which can kill bacteria. They infect a bacterial cell, multiply within the bacterial cell, lyse the cell, destroy the cell and come out. So initially he thought that his micrococci culture, which got contaminated with the natural mucus, there might have been a bacteriophage there. But later he showed that it was an enzyme which caused the destruction of these, which caused the lysis of the bacterial cells. He established that the lysozyme was an endogenous, meaning something which is synthesized innate antimicrobial in the body. The first discovery, remember, is in the nasal mucus of the Fleming. So it means that it is synthesized in human body. It is an innate antimicrobial which is present in all human beings. Later he found that this enzyme lysozyme was present in nasal mucus, in tears, and then he also discovered the same lysozyme in egg white, especially the amount of lysozyme, the quantity of lysozyme which he found was very high in hen egg white when compared to the quantity which is present in nasal mucus or in tears. The next question was, how does it work? If it is capable of lysing bacterial cells, how does it lyse? He found that the enzyme lysozyme cleaved, that is cut the purified oligosaccharides of N-acetylglucosamine containing a beta-1,4 glycosidic bond. If you remember the basic structure of a bacterial cell wall, you have an NAG-NAM linkage throughout, the beta-1,4 linkage. So if this N-acetylglucosamine, the beta-1,4 linkage is broken, the integrity of the cell wall gets broken. When the integrity of the cell wall membrane gets broken, the cell wall will, because of that loss of the membrane stability, the bacterial cell will lyse. And he also found that the oligosaccharides with more than 3 sugar units were also hydrolyzed, and the hexasaccharide was known to be the optimum length substrate. So lysozyme is capable of cleaving the NAG, which contains the beta-1,4 linkage, and also any containing the hexosaccharides, they were optimum length. Wherever this is present, the lysozyme could go and it could cause cleavage. And it was also seen that several families of lysozyme have been discovered. In spite of the fact that it is several families, all of them share the same property, the characteristic property of cleaving or cutting the beta glycosidic bond between the C1 of the NAM, N-acetylmuramic acid, and the C4 on the N-acetylglucosamine of the bacterial peptidoglycan, which means that the NAM-NAG bond, which is between the C1 of NAM and the C4 of NAG, it gets cleaved by lysozyme irrespective of whichever family of which member among the family of lysozyme you are using. Irrespective of which member you are going to use, all of them are capable of cutting open the NA1, the NAG-NAM bond, the beta glycosidic linkage, thereby disrupting the ball structure, causing the bacterial cell to lyse, which means that this is extremely effective in inhibiting bacteria. Further research has shown that you have more sources of these lysozyme like vegetables, insects, plants, fungi. All of these contain lysozyme, which means that it is a widespread natural antimicrobial prevalent in many species and many genera, or I would say spread over kingdom, because plants, you have the planta, they belong to the plant kingdom. Fungi comes as a separate kingdom. Humans, we come under separate division. It is present in insects. So it is spread. This natural antimicrobial is present over a wide different kingdoms and but the problem is the amount of the natural antimicrobial present which can be commercially extracted, that is less. So you have certain kingdom where you can observe it commercially extracted at a much higher quantity, whereas you have some where it is present, it is effective, but it cannot be commercially obtained. In humans, lysozyme is present in the human colostrum, in the mammalian tissues and fluids like milk, saliva, mucus, blood and tears. It is also present in high concentrations in macrophages, leukocytes, monocytes and neutrophilic granulocytes, which means that this enzyme that has a very important role in the human immune system. So when this gets stimulated, this forms the basic pathway in the immune response of organisms in reaction of infection and inflammation. So if this is effective in killing microbes by cleaving the beta glycosidic linkage, when the macrophages and leukocytes and monocytes get activated in the human body in response to an infection, along with the activity of these enzymes can also help in their even more way by trying to destroy the bacterial cells, helping in the reduction in the number of the infecting organisms. The most important source from which lysozyme can be extracted on an industrial scale is from chicken albumin. And if you talk about the food wise applications of lysozyme is normally added to baby formula so that it will break down more to help in the digestibility, and in also gastrointestinal treatments for the elderly. As you age, your GI tract gets compromised, the digestion as well as absorption of nutrients, the ability of the body to digest the food and to absorb the nutrients becomes less. So when we take care of geriatric people in what we call as geriatric nutrition, most of the products have a lysozyme added to it, so as to help the old people to gain nutrition easily, especially this is given as a treatment. Lysozyme is used in skin care to cure and prevent acne, bed sores, as well as in optical, dental and oral conditions. This lysozyme is also highly valued as a prescription and OTC remedy to treat headaches, colds and throat infections. OTC is over-the-counter medication, meaning this is not an antibiotic for which you need a doctor's prescription to buy. You can go to over the counter, you can buy this and use this to normally treat headaches, colds, etc. It is also used or administered to patients suffering from cancer because it is an analgesic, pain killing, and it has also been used as a potentiating agent in antibiotic therapy. Now, potentiating agent in antibiotic therapy, when you get infected with very serious bacterial infection, the doctor gives you antibiotic to cure it. But along with the antibiotics can be narrow spectrum, broad spectrum, act in multiple ways, that's a separate story. But if lysozyme is also given along with it, irrespective of whatever is the microbe, it will go and break the beta 1,4 linkage in the NAMNAG. So along with the potential antibiotic which is specific in action, the non-specific activity of the lysozyme will also help to reduce the microbial load, thereby increasing the rate of cure and making the patient recover better. So this is also used as a potentiating agent in the antibiotic therapy. Now, when we consider the food applications, Japan is a country where lysozyme is commercially extracted and it is also utilized as a trusted preservative for fruits, for vegetables, for tofu, bean, curd, seafood, meats, wines and sakes to naturally inhibit the growth of many spoilage organisms, to increase healthy shelf life and augment food safety. Now, because lysozyme is naturally present in multitude of kingdoms and you also have the lysozyme, addition of lysozyme is not going to create either allergicity or toxicity or any sort of adverse reactions. So this makes it very safe to use as a food preservative. The second aspect is, irrespective of whatever is the microbe you are talking about, it will go and break the beta 1,4 linkage, which makes it very effective. So whatever is the microbe which is present, for example, fruits contain certain specific microbes, the type of microbes in meat and seafood is totally different, what you see in wine is completely different, but irrespective of the class of the microbe you are talking about, wherever you have the beta 1,4 glycosidic linkage, there lysozyme will go and break. So you have a broad range of application against a wide variety of food products, all of which can be protected and preserved using lysozyme. And again, it is very safe because all of us have lysozyme we have it in your mucus, you have it in your nasal mucus, you have it in your tires, you have it in your saliva. So it's quite safe. it's not going to cross react or give you any other adverse reactions. And the food containing this, which has been preserved using lysozyme, is also, it is deemed according to the food safety rules and regulations, it's also considered safe. Many companies produce lysozyme and market it. You have, for example, you have Lysovin, which is specifically produced, which is, again, remember, lysozyme, you have a huge family with multiple members, but irrespective of the family, which one member you are talking about, all of them do the same type of reaction. So Lysovin is a specific product which is used to prevent the spoilage through the growth of lactic acid bacteria, specifically to inhibit lactic acid bacteria which cause spoilage of a fermented food or any food product. Then you have another type of lysozyme which is called as Lysolic R. This is again a lysozyme which is a safe and a natural antibacterial enzyme which prevents the late blowing in cheese, meaning the souring of the cheese towards the end, which completely changes the flavor and cause an off order and flavor to the cheese. This blowing, which we call as a late blowing, is because of the growth of Clostridium tyrobutricum. Okay, so this specific lysozyme Lysolac is capable of destroying that, thereby preventing late blowing by inhibiting the Clostridium botricum and increasing the shelf life. So, because this is natural and available, you can use lysozyme and replace chemical preservatives totally. And especially when we go for organic, we talk about the organic food products, there in the place of other food preservatives, the chemical food preservatives like sulfites and all the other components, we can use lysozyme in its place, even in the non-pasteurized food materials, and it would act as a good preservative. The company Biozootica produces another brand of lysozyme which is called as Lysobeer. This Lysobeer actually kill the beer spoilers, that is during production of beer when you want to have fermentation, but at the same time too much of fermentation by other microbes will produce an off taste. There we use this specific lysozyme to kill the spoilers, whereas by preserving the natural flavor and taste by maintaining the natural flora which is used. Now, with an example of the regulations, food safety regulations which is present in the US, I will try to explain how we specify that a product can be food grade. For example, properties and specifications of food grade lysozyme hydrochloride for use. This is the rules which are going to be present in United States. So, generally recognized as a safe, otherwise this is called as GRAS, G-R-A-S, we write it all in cap letters. It is called as the egg white lysozyme. As I said, the largest commercial source is from egg albumin, and this is from the extraction of the egg white. Then property of the enzyme, it is peptidoglycan N-acetyl muromoyl hydrolase, meaning it hydrolyzes the linkage whatever we are discussing about. So, this catalyzes the hydrolysis of the peptidoglycan in the cell walls of certain type of bacteria. Then what is the primary application? One, cheese. The primary enzyme targets are the germinating spores of Clostridium tyrobutricum, an organism responsible for the late bloating of certain cheese varieties. Clostridium is a gram-positive spore forming organism. This is not a pathogen, but you have the other very well known species of Clostridium botulinum, which is also a spore former, which is a extremely lethal pathogen. And here, as you know, the spores are dormant and they are capable of being there, meaning not getting killed in spite of the multiple methods because of the specific production. So, for these to be killed, you need either highly potent antibiotics or specific methods. So, this lysozyme, especially when the Clostridium spores are germinating, okay, this lysozyme is capable of going and breaking down the walls, preventing the germinating bacteria or lysing the germinating bacteria, thereby inhibiting its growth in the food product. That is how that's its mode of action. Now, use level, meaning how much quantity of lysozyme can be used safely in the product. So, this is specified according to the current good manufacturing practices, especially in cheese. So, for any food product you have GMP, that food additives this much has to be added, food preservative this can be added. So, for each particular level, for each food product you have a good manufacturing practices which are specified. So, that lysozyme can be used depending upon that particular GMP. The name of the enzyme is lysozyme hydrochloride, and when you add, as you know, all of them require a label. So, in the label what do we write? In bulk and packaged foods that contain cheese manufactured using egg white lysozyme, it will include the usual name egg white lysozyme on its ingredient label, and this term indicates that it is food grade. It is manufactured from egg white lysozyme, it is commercial ingredient, it is food grade. So, all these are the conditions which have to be followed if lysozyme is used for preservation of particular food. This is the picture, this is how Lysolac looks like. This is taken from the website Bioceutica, which produces different types of lysozyme for particular type of food products. So, as we saw, majorly we discussed about 5 types of antimicrobials: nisin, tylosin, subtilin, natamycin and egg white lysozyme. We saw the potential food applications of all these, what is the mode of action, how these work. Similar to these specified products, you have a huge grade of other types of natural antimicrobials also with potential applications in food industry. Scientists are trying to make a list of them, trying to find a mode of action and also to find out the stability, the pH sensitivity, the temperature range over which they will function, and what are the types of food which can be effectively preserved using these natural antimicrobials, because anything natural tends to be more welcome rather than using chemical substitutes. At the end of this session, we discussed on natural antimicrobials and how effective they are. We also have there are other natural antimicrobials also which are potentially equally useful. We need to further evaluate, analyze their action, and they can also be potentially brought into for use in the food industry. Thank you.